warm welcome to today's ArangoDB online meetup. Today with us is Tariq, and my name is Jana. I'm part of the ArangoDB's marketing team. I will just do a very quick introduction and then give the word to our speaker. So today's meetup is part of the ArangoDB Community Pioneers Initiative. In case you haven't joined any of our previous meetups, quick introduction. This is a project that we started together with the community. And Tariq, I'm so sorry, could you mute yourself? Okay, thank you. I hope everybody can hear me. So just to jump back, there will also be a recording of this talk. Of course, we will post the recording on the meetup.com and send you a follow-up with the recording. So going back to the ArangoDB Community Pioneers Initiative and the quick introduction. The way you can become an ArangoDB Community Pioneer, I mean, basically it's already all of you guys who are supporting our open source project all around the globe. We are really, really grateful for all the contributions that you are doing on a daily basis. You can get involved with the program by doing a talk in our online meetup, like what we're doing today, or you can also join your local community online and perhaps do a talk about ArangoDB there. You can write a short blog post, a tutorial, or a use case about something that you've done with ArangoDB or how ArangoDB helped you solve problem A and which queries you used and what is your little use case that you have been playing around with. Or you can also simply tell others about your experience and what you think about the database. You can write a small thing on Medium, you can write a review on G2Crowd, or also just add ArangoDB as a skill on LinkedIn. If you would like to contribute and if you would like to get involved, so please write me a small note. There's my email address, so you can ping me on Twitter or on our community Slack. Also, as I already mentioned before, my name is Jana. So I would love to hear from you guys and just ping me anytime. But not to keep you waiting any longer from today's talk. Today with us is Tariq, who has been our contributor and a community member for quite a long time, actually. Tariq today is going to talk about machine learning and immunology and how ArangoDB actually saved his life. I am super, super excited to hear the talk of Tariq today. He's joining live from Montreal. Not to keep you waiting any longer, warm welcome, Tariq. And let's get started.
Thank you, Jana. Okay, can everybody hear me? Can you just hear me?
Yes, the voice is really good.
Thank you. Good. Perfect. So thank you guys. Thank you. Big thanks to ArangoDB for inviting me to talk. So this is a story of my life, and also one of the stories why I got convinced that ArangoDB is the best database that exists. So a little bit about myself. I started for my background. I did a master's in machine learning, and basically what I had to do is to make a virtual drummer, and I had a curated data set. Someone else had perfectly curated a set of MIDI files that I could use. But then I transitioned to a Ph.D. in Bioinformatics, and the goal was to find better vaccine targets, and this was completely different. It was what I call the fog of war. So completely different way of doing and applying machine learning. And so this is the story of how I actually used ArangoDB to clean up, to get myself out of this fog of war. So this talk is called Machine Learning, Immunology, How ArangoDB Saved My Life. And so thank you very much, ArangoDB, for saving my life. And my name is Tariq Daouda. So data is very, very important in machine learning, and usually today it's the most perhaps the most important thing that you have when you do data analysis and computer science. So if you look at the pyramid, you have data. So data is the bottom. Data is the most important thing. And then on top of data, good algorithms, and you can critical to get this part right. Machine learning context, like what I had to do for my master's, usually have a data set that's been already created, and you iterate this way. So you have some machine learning hypothesis, and then you do experiments, and then you get something, you attain your goal, which is, for example, their accuracy. And this part is always changing, always making new hypotheses, and always do testing them to see if they work or not. But in life sciences, things are a bit different. Your data set can change. You also have two sets of hypotheses. You have the machine learning hypothesis and you have your biological hypothesis and all of them actually influence the kind of experiments that you're going to do. And then you also have the research question that can change because as you discover new things, the research question themselves become different and evolve. So this is what creates that fog of war. So for the data set, for example, data set that I had to use was not yet generated, was not yet made when I started the project. You often have to deal with many types of data. You always have to deal with new hypotheses coming from your colleagues, that do biology. You always have to deal with multiple file formats for DNA sequences, for genetic mutations, and various things that usually are in CSV with different types of columns. And so it's really a complex, much more complex setup for doing machine learning. So what I did in the beginning, just I just used like the normal naive approach. So I had my raw files in a bunch of different formats. And then I wrote a script that basically took all this and made me a nice data set that I could put into my machine learning algorithms. And the problem with this approach is as the weeks go by, the difficulty to maintain this script becomes exponentially higher and higher and higher. And also the slowness, the script becomes slower and slower and slower. And this is because you have so many things you deal with. Each time you have something new, you add it into that same script. And so that produces bugs. It's very hard to maintain. You have reproducibility issues, and you can also draw false conclusions. And this can be, within a research context, this can be very dramatic things to, you should avoid having false conclusions. So because we are in this context that where everything changes, we need a method that actually allows you to move fast, to test as many hypotheses as fast as possible and as cheaply as possible. You also need something that's maintainable. And first approach, the naive approach, was not cutting it for me. So which brings us to the data handling, and basically it's do-it-yourself ETL. ETL stands for extract, transform, and load. So for that, I decided to use a database. So you have many choices that you can choose from. You have the standard SQL databases, and you also have this huge set of NoSQL databases that you can choose from. So I did a review of all this, trying to find the best database for my project, and I ended up using ArangoDB. So why did I use ArangoDB? Because first of all, ArangoDB is very, very fast. It's so fast that sometimes I forgot to add the indexes that I haven't noticed that I forgot to add the indexes to the database. So ArangoDB is very fast. ArangoDB is also multi-modal, and this is a very, very important aspect for me because in this context where data keeps coming to you in different formats, you don't really know firsthand which will be the best model to schematize and store your database. So with ArangoDB, you don't have to make a choice when you choose database. You can either, depending on data that you have, you can model it as a graph, as a document database, or as a key value database, depending on your application. ArangoDB also has an amazing documentation, especially for the HTTP API, because I'll talk about it later, but I had to write a Python driver for ArangoDB, and the documentation of ArangoDB was exceptionally good. ArangoDB also has a query language, AQL, which is basically ArangoDB's SQL. And AQL is a very nice tool. It's very easy to understand, and it also allows you to write very compact queries in a way that's very easy to understand. And finally, ArangoDB has a web interface that allows you to test queries and also facilitate collaboration with non-technical colleagues, because you can just send them the link to the database that they can look at the documents themselves. And also, it also facilitates exploration a lot. So the first thing that I had to do is to organize the data. So this is what the whole thing looked like. So I had 55,000 peptides, 33 MHC1 allele, 18 subjects, different gene expressions, the whole human genome, and the whole human proteins. So the first thing is to figure out how to organize all these things so they can fit together in the same database. So with ArangoDB, because you can model your data as a graph fits perfectly for this kind of setup. So you have 18 subjects. These subjects have genes, and these genes have expressions. So you can use an edge between subject and humans in the human genome. So you can just point out an edge that links the subject to a gene with a given expression. And also in the human genome, you have genes. Genes have different versions that are called transcripts, and then these transcripts are translated into proteins. So you can just mix into the same document the genes and the protein and the transcript. So you can just merge them together. So now everything becomes connected to the subjects thanks to the edges of gene expression. So for the peptides have been identified from the cells of the individuals, and these peptides bind to specific molecules that we call MHC1 alleles, and they have binding scores. So yet again, we could use edges to connect the subject to the peptide. So because the peptides come from the subject and they bound the specific MHC allele of the subject, we can just connect them with an edge that has a value binding score. So we know that this peptide was found in this subject with binding that specific MHC allele with this specific binding score. And then these peptides, they come from proteins. So yet again, we can use an edge. And this edge also encodes the position where the peptide was found in the protein. So the peptide is a small bit of the protein. And in this way, we can store where the peptide exactly came from. So all of a sudden, this makes much more sense than what I had before here. Now everything is nicely connected and nicely parsable. So the goal now is to take this schema and put it into ArangoDB. So this is where the extract, transform, load paradigm comes into being. So I had different files. So the extract, you will extract the data from the files and then send them to ArangoDB. Then you have the transform. This basically a filtering step that will put all this data into a nice pickle that I can load into a machine learning algorithm. So the extract is a little bit more complicated than just extracting the data. So you have this file. The first thing that you have to do is you have to parse the different files in the different file formats, and then the next thing you have to do is to harmonize everything. So you have to harmonize column names, you have to harmonize the values. Sometimes you have undef, sometimes you have anna, sometimes you have nothing, sometimes you have a question mark. It depends. These files usually have been made by different people at different times using different nomenclature. So you have to harmonize it. And this harmonization step is actually one of the steps that made my first big script huge. So you have to be very careful with that and not forget about this part. And then you have a validation step. And the validation step makes sure that the harmonization has been made correctly. And then you can store it into ArangoDB. So everything that's linked to genomes and proteomes, I already had written a Python package called PyGeno. So PyGeno already takes care of the parsing of the harmonization. So we just give it like this bunch of FASTA, VCF, and GTF files inside the genome. Pagina gives me nice sequences, DNA sequences, and protein sequences that I can directly store into ArangoDB. But for everything else, the CSVs, basically I had to write the parsers and also the harmonization. So it's always best to separate the harmonization, to have different scripts and not just one script that harmonizes everything. But you have different, for different, every set you can have a script of that harmonizes the CSV, and then you get, the goal is not to store it directly into ArangoDB, but to actually store a version that's a clean CSV. So the clean CSVs, the CSVs, and the clean CSVs are the clean CSVs, how they've been harmonized. They all have the same name column. They all have the same values in the columns. And of course, you have to keep these ones somewhere saved just in case you need to go back. But all the rest of the analysis will be done on the clean CSVs. Yeah. So for the validation and the storing, I used Arango that I actually wrote for partly for this purpose. And so the nice thing, Arango is a Python driver for ArangoDB. So it allows you to connect your Python script to an ArangoDB database. But the nice thing about Arango is that you can set up a bunch of validators for your fields. So, for example, for this collection transcript, a transcript in that collection, you can trigger the validation to happen on save and on set. So every time the document is saved, the validation will be triggered. On set, every time a new value is set, the validation will be triggered for that value also. You could set up allow foreign fields to false, which means that only these fields listed here will be accepted during the validation process. And then for every one of the fields, you can give a set of validators. So the name has to be there, it cannot be null, it has to be a string. The length cannot be null either, it has to be indent, etc., etc. So it allows you to declaratively set up your validation, and that cuts enormously in the ifs and loops that you have to do in your code, especially if you have different files that all have the same kind of data that you have harmonized, and then you want to make sure that the harmonization has been made correctly, you can set up a set of validators here that actually make really sure that the values are the same, and you have ints where you should have ints, and the ranges are exactly the same, etc., etc., before you can store them into ArangoDB. So now the transform part. So the transform part is actually the critical part, because if you look at the extract transform load paradigm, in this case, the extract is very static. So you have your data, you don't change it that much. You only change it when you get a new CSV from your collaborators. This doesn't happen every second, like maybe every few weeks or every week. You get a new set of data. So this is fairly static. The loading is also very easy, because if you can get a JSON, it's very easy to transform the JSON into a pickle. So also the scripts for loading are fairly static too. But what really changes a lot is the transform part, the filtering, because this is really what's influenced by the different by the new hypotheses that you're going to generate. And so basically, by filtering, I mean for example, what gene expression what threshold for gene expression should we use? What threshold for binding score should we use? Do we have to only consider, for example, the peptides that have been linked to 3 people, and remove the ones that have been only linked to 1 person or 2, and things like that. So you have so many questions, and all these questions are basically, you cannot answer them if you don't filter the data first. So the filtering is really here the critical part that changes a lot. And this is when ArangoDB really shines, because I can express, using AQL, I can express very complex queries very, very easily. So for example, I can ask for all peptides that come from transcript with a minimum expression of 10 to the 5 and a minimum binding score of 1000. And I want the peptide sequence, I want the binding score, I want the protein sequence, I want basically a lot of stuff. So if I had to do that like the traditional normal way without using a database, I would use pandas and Python, and my code will become more and more complex, and I will have to deal with the manual caching and indexing of huge files, because for example I would have to have the whole human proteome index somewhere and the whole genome index somewhere else, and this this makes for huge files, long waiting times, and very complex code. But with ArangoDB, I can just express all that with a simple AQL query, and this AQL runs in milliseconds, and this is super flexible. And I get exactly what I want and what I need for running my experiment. And also, what's really nice is these AQL. AQL is easy to write, but sometimes you need to test the queries before you can you can really apply them. So ArangoDB has this nice interface where you can just test the queries, and you can even download the JSON after you query. So this makes it really easy to write the right queries that you need. And also, which is really nice, ArangoDB has this nice graph visualization. So um, you can at any time you can open the graph visualization and look at how your data is organized, is everything has been saved correctly, and you can also say that send that to non-technical colleagues, and they can play with the data and they can extract the data themselves. So for the transform, what I used is basically I would write each time I would like to make a new type of data set. I would just write a script. It's always the same with the same architecture of code. It's a very simple script with an AQL query that I can launch using a very simple command line interface. So this script will go around ArangoDB, get the JSON, and then save that JSON into a pickle, and then give it and then I can give that pickle to the machine learning algorithm. So this is extremely easy to maintain, and also it makes my results extremely like super reproducible, because what I have to do is to store in that pickle. What I do is I store basically the arguments that I gave to the command line. So if I ever want to regenerate the same pickle, I can, and I can do it in a simple line of code, usually a few seconds, and I have exactly the same data the same pickle that I used to have. And also this can be done with very few lines of code, and always the same type of line of code. So it's just an AQL query, you get the data, you save it, you get the JSON. JSON is basically a Python dictionary, and then you save that Python dictionary and you give that directly to your algorithm. So very easy, very straightforward, and very easy to maintain. And this actually has become very critical, because this project has been going on for like 7 years, and thanks to this approach, we were able to basically reproduce results for any given time, and this is something to really do good science. So in conclusion, with this method, the fog of war is gone. You have a system that is very fast and very reliable for storing, managing, extracting your data, getting your data set for your machine learning algorithms. This system takes less time for maintaining, and which gives you more time for science. It also allows you to move quickly and test new hypotheses, and it's much cheaper to also test new hypotheses, which means that you're more likely to test them also, and that gives you better chances of making discoveries, which is very important when you're doing science. And also, it significantly decreases your frustration, and it also increases your quality of life, because you spend much less time trying to debug and maintain very convoluted code. So, and just to finish, a similar system, we're using now a similar system. We started a project for trying to speed up the research to find a COVID-19 vaccine, and we're using a similar system to actually store our data and to make an open API that people can query, and also an interface that people, like a portal, that graphical interface people can use to navigate the data. And it's all basically the same similar system as the system that I've just talked about. So this website is called epitops.word. It's still not on, but we're working very hard to get it to get it to the public as fast as possible. And of course, we're using ArangoDB, and ArangoDB has been very useful and very nice by providing us with free cloud hosting on Oasis, and it works perfectly. So it's great. And that's it. So that's the end for the presentation. And you can also use ArangoDB to store your experiments, metadata, and etc. So I haven't talked about this part, but there's something now called ArangoML that you could use to actually store your metadata. It's a new stuff, and it's really cool. So I encourage you to give it a try. And that's it. Thanks. Thank you for your attention.
Tariq, thank you very, very much.